Hi, everyone. My name is Travis Yates, and I'm a field application scientist with Personalis. The topic of my presentation will focus on advancing biomarker discovery and how we at Personalis are working to improve tumor immunogenomics by taking a comprehensive approach to understanding malignant disease. At Personalis, we've really recognized that there are several issues plaguing biomarker advancement. One of the first of these, and maybe the most important, is the struggle with limited and precious tumor specimens. Now, on the left, I'm showing that for a full evaluation of a sample's potential, a multitude of different specialized testing needs to occur. This is often done using different technologies with multiple vendors. This can lead to a number of issues that compound, things such as time of analysis, complications with data integration, and so on. Now, at Personalis, we've taken these factors into consideration, and we really work to create a more efficient platform and process that keeps sample sparsity in mind while providing a multitude of high-quality data. Now, our process starts with the initial receipt of limited sample volume, followed by dual extraction of DNA and RNA, which is then processed onto our platform to produce a rich array of analytics. And this makes up our ImmunoID Next platform. Now, with ImmunoID Next, we have developed an approach to enable comprehensive characterization of the tumor genomics, but to also allow for interrogation of the immune component and tumor microenvironment from a single tumor sample and matched normal. Now, we maintain a sample sparing approach and can work with as little as 4 FFP slides to derive DNA and RNA, which we interrogate from multiple biomarkers. To give you an idea of some of the challenges in conventional sequencing, here in the upper left, I'm showing an example of a gene sequenced with a standard exome kit at high depth. Now, this is highlighted in blue, and as you look across the gene, what you're going to note is that there are areas that I'm calling peaks and valleys in sequencing coverage. Now, these are areas that are sufficiently covered, the peaks. And while there are other areas that are suboptimal, and these would be the low-lying valleys. Now, even though this gene was sequenced at a high depth, it fails to cover the entire exonic region. And if I'll draw your attention to the red dot, now if there was a variant that was residing in this region, it would go completely undetected and be missed using the standard whole exome sequencing assay. Now, at Personalis, we went and we systematically identified where these gaps and inconsistencies are. And we've designed probes to target and substantially augment these areas. This technology allows for accuracy and content enhancement, or as we call it, ACE. Now, we've done this supplementation to all 20,000 genes with ImmunoID Next, and that is highlighted in the green behind the blue graph, and that's that same gene sequenced with our ACE technology. And as you'll note, we've been able to fill in those gaps and low coverage areas. Our ACE technology is also the core of our transcriptome assay, which takes an exome capture approach using our ACE probes positioned at all 20,000 genes. Now, we find that this capture approach helps us improve performance of RNA derived from FFP samples over other methods like poly-A selection or ribodepletion. We can achieve uniformity in coverage and increase sensitivity, and this is particularly important as we use our RNA data to derive multiple IO biomarkers. Unlike a traditional exome and transcriptome assay, the design of ImmunoID Next has unique features. We sequence at an increased depth for both the exome and the transcriptome. In addition, we've also had that augmentation of all 20,000 genes. We've added a unique boost to actionable cancer-related genes at 1,000x coverage, which allows for profiling. Additionally, we've boosted coverage of IO regions such as the TCR and BCR genes from the transcriptome. Now, we've improved coverage of HLA genes. We've placed probes in microsatellite regions to detect instability. We target 7 of the most common oncoviruses, and we've really worked to create a comprehensive assay for immunogenomics profiling. In addition to continuing to advance our platform, we've also co-developed analytical solutions to help to make sense of all of the data produced by ImmunoID Next, and we report on multiple biomarkers. Now, we've built our own internal tool for HLA-related features, whether this is typing from the germline, somatic calls in HLA genes, or loss of heterozygosity detection. We report on TMB, and we've also developed a neoantigen prediction pipeline. Our repertoire ID enables the analysis of the clonality and diversification of the array of TCRs present in the tumor microenvironment. We have oncovirus detection, and as I mentioned, we report on 7 of the most common oncoviruses, but we can actually delineate between their various genotypes. We have microsatellite instability characterization. And finally, our immunogenomics ID provides an overview of the tumor microenvironment and critical genes involved in the tumor and immune biology, such as the adaptive and innate immune response, immune checkpoint modulation, antigen presenting processing machinery, and so on. Now, in this slide, I'd like to take you through a melanoma case study that we did in collaboration with Dr. Shipwan Zhang at the Innova Cancer Institute. So let's just start with a general study overview. Now, we obtained samples from 100 patients with late-stage unresectable melanoma who had received an anti-PD-1 therapy. Now, this was either Keytruda or Opdivo. And we looked at the first 51 patients in this set whom we had clinical outcome results. Now, here, clinical response was determined based off of risk criteria, and this was best overall response and progression-free survival. Now, we had responders and non-responders. The two groups were matched in terms of demographics, age, gender, and other factors. Now, the baseline tumor sample and paired normal were sequenced and analyzed in our lab, and we explored readouts from ImmunoID Next analytics and plotted several metrics to identify mechanisms of resistance and response profiles. Now, one of the first things that we looked at was we calculated neoantigen burden in this cohort. And what we found was that it's significantly associated with both response and improved progression-free survival. However, it was interesting to us because we observed unique profiles that from outliers that we were interested in exploring through immunogenomics characterization. And that's what I'd like to walk you through in here in the next just few seconds. Now, the first that we're looking at was actually was in the responders group. This was a partial responder, but it had a relatively high neoantigen burden. Now, what we found is that each of the HLA class 1 genes, A, B, and C, in this partial responder had 2 distinct alleles from typing the germline. However, by looking at the tumor, we found that there were chromosomal deletion resulting in the removal of 1 allele of all 3 of the HLA genes. Now, this loss of heterozygosity event can potentially be detrimental to the number of neoantigens that can be presented for immune surveillance in this particular individual. And it could explain the partial response to checkpoint blockade. Now, the next patient was in the responders group, and this was in the non-responders group. And now this patient also had a very high neoantigen burden. However, in this patient, we detected somatic mutations impacting 2 HLA class 1 genes. Now, both mutations had a high allele frequency and a high variant effect impact, suggesting that they may be affecting the function of the HLA molecules. Now, our final patient was once again in the non-responders group, and we observed a somatic frame shift variant in a large portion of the reads, within the beta-2 microglobulin gene, which is a key component of the antigen presenting complex. Now, this frame shift variant potentially limits the correct HLA class 1 folding and antigen presentation and could be, once again, a mechanism of resistance. Now, if we look at the neoantigen burden, again, but we take into account alterations impacting HLA genes and antigen presenting machinery, we can produce an adjusted neoantigen burden as a composite biomarker. Now, by doing this, we observe a tighter correlation with clinical outcome and better separation of the groups. And we've termed this NEOPS. And this is a composite biomarker that is an output from ImmunoID Next. Over the next few slides, I'd like to highlight our next liquid biopsy and really describe how it complements our ImmunoID Next platform. Now, you've already heard about ImmunoID Next and how it can provide enhanced picture of the solid tumor. But now I'd like to discuss with you how our liquid biopsy strategy fits with Next. You'll see that our strategy is built around the concept of going beyond and providing a much more comprehensive view of the tumor using our liquid biopsy. And our strategy really starts where we have a full picture of the solid tumor through immunogenomics profiling. But in addition, we now have an exome-wide look at the plasma as well. Now, our next liquid biopsy leverages our DNA exome footprint of our solid tumor offering, interrogating all 20,000 genes. And I'm just showing here that, we pair this with our ImmunoID Next, and we're able to look at this at, baseline, but we're also able to then look at liquid biopsy samples at different time points, possibly when on treatment. Now, this is just a schematic of our assay design. And with our innovative approach, it enables us to achieve a high-level performance at exome scale. Now, just as our ImmunoID Next, our liquid biopsy assay uses our proprietary ACE technology to fill in those sequencing gaps and coverages missed in more conventional approaches. Now, we achieve 2000x depth across the entire exome and roughly 5000x in the boosted coverage of those actionable cancer-related genes. Now, for this assay, we wanted to provide an extraordinarily high depth for exome sequencing, coupled with our comprehensive bioinformatic approach to yield excellent performance. Now, one of the important pieces here is that we really see Next liquid biopsy as a complementary to our ImmunoID Next. And when you're really trying to build a comprehensive view of what's happening in the tumor, it's ideal to have both the solid tumor and the liquid biopsy because there are elements of the biology that you can only see in one or the other. So if you're looking for full characterization of the tumor, the tissue remains important because you can assess, various information. You can look at, the tumor microenvironment. You can robustly interrogate the RNA for gene and immune expression signatures. You can explore the TCR alpha and beta clonality. And so, we think that the tissue sequencing remains a gold standard because of the information that it provides. However, with that being said, our liquid biopsy is important because with it, we're able to understand the inter and intra-tumor heterogeneity. And we're really addressing, this problem more than, say, with the whole exome, as opposed to, say, 10 to 500 gene panel. And of course, with liquid biopsy, you have a convenient and high compliance way of acquiring samples. So if you put these two together, ImmunoID Next and Next liquid biopsy, you can get a better picture of what's happening in the context of the disease. Now, on this chart, I'm really just demonstrating the number of genes that are interrogated by Next liquid biopsy in blue. Now, this is compared across various key areas of tumor biology to two of the gene panels. That would be orange and gray. Now, what I hope you can appreciate is really just the increase of information in areas like, say, the adaptive immune response, antigen-presenting machinery, chemokines, cytokines, and many others. These are overall, very exciting in that we're providing further capability to our partners. Now, what questions can whole exome sequencing from the plasma answer? Now, it can provide an enhanced assessment of tumor spatial heterogeneity by providing a broader view of the potential driver mutations, detection of subclones that can confirm, intrinsic therapeutic resistance. It can allow for monitoring of clonal and subclonal dynamics in response to therapy. And finally, it can provide a method for interrogating acquired mechanisms of resistance. Now, at Personalis, we've really worked hard to become partners throughout all phases of clinical development and translational research. However, what I hope I've been able to do today is to highlight how our ImmunoID Next platform and our Next liquid biopsy can act to comprehensively support our pharma partners and provide a great deal of insight for their patients in their trials. In addition to that, we have a number of different offerings and avenues. And we try to support our, genomics partners across the entire drug discovery continuum. Thank you. And we'd love to hear from you if you'd like more information on anything that I've talked about.